Okay, so welcome back from your break. Let's take a look at this question. So why are bacteria considered to be the most successful organisms on the planet? Well, in terms of sheer diversity, in terms of biomass, in terms of numbers, in terms of effect on the planet, these guys win hands down. So they're the first cells to emerge from the abiotic soup of chemicals. They create huge changes. I mean, just look at the oxygen revolution. Remember, oxygen does not become a significant part of the atmosphere until around 2.5 billion years ago, approximately. And it's the product of cyanobacteria, so it's the product of bacteria. And that would change the total directory of life completely, because you had three options: adapt, run away, or die. And in the case of most organisms, die was the only option, become extinct. But it would lead to eventually cell respiration and eukaryotic cells and multicellular organisms and fungi and animals and plants and algae and all of that as a direct response to oxygenic photosynthesis evolving in cyanobacteria. And you can't say that these have not been significant. There are so many ways in which bacteria have been significant. They're decomposers. They act as symbionts to allow organisms to grow better. They have huge impacts on the environment. And the final point, if they disappeared, life would shut down. There is no way that life as we know it could exist without the presence of prokaryotes. The nitrogen cycle decomposes other chemical cycling that they do. Without that, life as we know it would cease to exist. In fact, life period would cease to exist without prokaryotes. And in case you think, well, this doesn't really apply to me, I hate to be the bearer of bad news, perhaps, but your body contains somewhere around 10 trillion cells. There are somewhere between 10 and 100-fold more bacterial cells living in your body, which begs the question, are we really a eukaryotic organism or just a convenient container for bacteria as a culture place for bacteria? Interesting question to ask. Anyway, onwards and upwards towards the end of this presentation. So what is the impact of prokaryotes on humans? Well, as I said, about 50% of all human diseases are caused by bacteria. So we know of a number of diseases that are caused by bacteria. I'll show you a list of some of the more interesting ones shortly. So Lyme disease transmitted by tick bites, the deer tick particularly, it's a spirochete. It gives you this bullseye pattern radiating out from the site of the original bite. Some people say that we don't have Lyme disease here in Florida. In fact, we do, particularly up in North Florida, but there have been some incidences down here in Central Florida. What is most disturbing about Lyme disease is that it takes a long time for antibiotics to eliminate it. It can be 12 to 18 months of consistent antibiotic treatments to get rid of the spirochetes. They seem to be very resistant to drug treatment. I've talked about Koch's postulates when I was talking about the Helicobacter. Here are the postulates laid out for you, the 4 postulates in total. Find the pathogen in all individuals who have the disease, isolate the pathogen and cultivate it as a pure culture. Use that culture to infect healthy hosts. That host then comes down with the disease and re-isolate the pathogen from the now diseased host. Those are the postulates, and as I showed you with the Helicobacter, the 2 investigators followed these postulates to demonstrate the Helicobacter is in fact the causative agent of the majority of stomach ulcers. So here are some other human diseases and the bacteria that cause them. So you can look through this. You're not going to find this in the textbook. This is a list that I put together myself because I thought this was kind of interesting. So you can follow through this. It's also in the lab manual. We published this in the lab manual as well in the bacteria lab. So you've probably already seen this because you've done the bacterial lab. But it's interesting to look at the various types of bacteria that can cause different diseases. There's one or two that I might highlight. I think Vibrio down here, Vibrio parahemolyticus. This is the bacterium that you contract by eating raw oysters. It gives a particularly virulent form of food poisoning that can be very lethal. It's the reason why every bag of oysters that's harvested in the United States carries a tag indicating where and when it was harvested in case someone comes down with a case of food poisoning, so they can track all of those shipments. It's also the reason why periodically they shut down the oyster beds in Apalachicola Bay, because the primary source of Vibrio contamination is untreated human sewage. And several rivers that run through Florida enter into Apalachicola Bay. And one of the things that you will learn about oysters when we get to the animal kingdom later in the semester is that oysters specialize in filtering the crap out of water in both a figurative and a literal sense. This is the reason why they become contaminated with all sorts of bacteria. Yersinia pestis, the bacterium that causes plague, or at least we think it's the bacterium that causes both modern plague, which we know for sure, but we also think it's the bacterium that caused the plague back in history, starting, as far as Europe is concerned, around 1330 and coming forwards from there. Clostridium. The Clostridium species are particularly nasty. So Clostridium botulinum gives rise to botulism, a particularly nasty form of food poisoning associated with poorly sterilized canned food. If you see a can that is bulging at the ends, throw it away. Do not even bother to open it, because that bulge is telling you that something on the inside desperately wants to get out. And it is carbon dioxide produced via anaerobic respiration by Clostridium botulinum growing inside the cans. It's an ideal place for botulinum. It likes an aerobic environment. It likes to have plenty of organic material. In a can, it has that because the canned foods are not sterilized until the cans are sealed. So if that sterilization doesn't take place properly, it provides a good environment for botulinum to grow. Ironically, the botulinum toxin, which is one of these lipopolysaccharides that I was talking about earlier, in dilute form is the active ingredient in Botox injections, which is the reason why, if the Botox injections are given by someone who doesn't know what they're doing, it can be lethal, and people have been killed by unlicensed individuals giving Botox injections. So the Clostridiums are gram negatives, and they do produce these lipopolysaccharides, and they produce some particularly nasty lipopolysaccharides. Tetany, of course, is what causes tetanus. You are up to date on your tetanus shots, right? Every 10 years, you need a booster. So basically, the pathogenic prokaryotes can produce two different types of toxins. This is toxin biology 101. So you've got exotoxins that are secreted from the cell. Remember, exo, out. And endotoxins, these are the toxic materials, the lipopolysaccharides that are produced by gram negatives. So in the case of the endotoxins, you do need the active bacteria to be present. In the case of the exotoxins, not necessary, because they've been secreted from the cell, they're in the environment, you come in contact with them, you get sick, you may die. And we talked about anthrax as a weaponized bacterium. Other types of bacteria can also be potentially utilized as weapons. What is somewhat scary is that because of the ease at which we can genetically modify bacteria through the plasmids, there is the potential for creating truly weaponized bacteria through genetic engineering. And that's kind of scary. That's why the Army has Fort Detrick. And I imagine that's why Russia has its own biowarfare campus in Siberia as well, for that purpose. We also know that there are ways of utilizing prokaryotes for research. So the book introduces you to the CRISPR-Cas9 system. This is actually a two-part system. CRISPR and Cas9 are two different mechanisms that bacteria have evolved to protect themselves against viral infections. So the CRISPR part of the combination was identified first. And I recently listened to a podcast with the researcher who identified the CRISPR mechanism. She then encountered a colleague that had been investigating the Cas9 protective system, and they realized that the CRISPR-Cas9 as a combination then allowed for gene editing, not just in bacteria, but in any organism, because the CRISPR system uses a guide nucleic acid sequence to identify a gene of interest that the guide nucleic acid base pairs to, and then the Cas9 proteins can do cutting and editing. So this is why the CRISPR-Cas9 system is so interesting, and why people are now actively investigating it, because it really does allow you to selectively edit a specific gene using the guide DNA to take you to the gene of interest, and then use the Cas9 system and the guide and an intact copy of the gene to edit the defective genes to create a working copy of the gene again through the CRISPR-Cas9 system. It's a really cool system. Don't have time to talk to you more about this. Then prokaryotes have been used for a number of different biochemical treatments. So here you can see a man spraying a nutritive solution on rocks up in Alaska when they had a major oil spill up there. The oil contaminated the rocks. It's well known that there are bacteria in the environment that metabolize oil, because oil seeps out of the ground worldwide in small amounts. So bacteria have evolved to utilize the crude oil. And this was an attempt to try and activate those bacteria to break down the oil spill. It did not prove to be successful. These bacteria did not break down the oil spill. However, when we had the major oil spill around the oil platform in the Gulf of Mexico a few years ago, as part of the research post oil spill, once they shut everything down and capped the well, was the discovery of at least 6 new species of bacteria that metabolize crude oil that were found around the wellhead deep in the Gulf. These were totally unknown up until this point in time. So as much as the oil spill was a tragedy because people died and it contaminated the Gulf Coast, something good may come out of that catastrophe, because these bacteria are now being investigated as potential bioremediation, because they are much better than breaking down oil than these rocks, these bacteria in Alaska. Bacteria in this central image are producing plastic. These globules are actually a bioplastic that these bacteria produce. And bacteria are really good at making organics and making polymers, and these ones have been engineered to increase the production of a naturally occurring plastic polymer that they produce. And what is great about this is that it is biodegradable. What does biodegradable mean? Let me write a definition up here. So biodegradable. Let's try that again. And to work this time. So biodegradable. And naturally occurring prokaryote exists that will degrade it or will break it down. And as you've seen, bacteria are really flexible in what they will utilize as a source of energy. And so this is why bacteria are of interest for all sorts of bioremediation activities, I guess. But this biopolymer has now taken on a commercial value because it is used in a number of different plastic containers. So a number of those clear plastic containers that you can buy food in at the supermarket, they are actually built out of this biodegradable polymer that's made by bacteria. Some of the initial biopolymers that were used had an unfortunate byproduct in that they were UV sensitive, and so they would break down in sunlight. But some of the more recent ones that have been identified are actually UV stable, but they can be broken down by other bacteria. They are biodegradable, so they make a useful container because the plastic that they contain is produced by bacteria and is broken down by bacteria once it returns to the landfill. The left-hand image is an indication of wanting to use bacteria to make biofuels. Because they can make all sorts of organic molecules, some of them can make biofuels that can be used as a supplement to existing fossil fuels. So there is a biodiesel. Willie Nelson is actually a big proponent of this. He sells a version of biodiesel at some gas stations that he owns down here in the south. It's called Bio Willie. It's a biodiesel that is mixed with regular diesel fuel to supplement it. And it is produced by bacteria. And bacteria can also produce ethanol or indirectly be used to produce ethanol, because bacteria also, or some archaea, not so much bacteria, but some archaea, have the ability to break down cellulose, and they can break down cellulose to glucose that then can be metabolized to ethanol through fermentation by yeasts. And so it's an alternative way of making ethanol other than fermenting starch from corn, which actually takes corn out of the food chain. So there are a number of different ways that we can use prokaryotes to our own advantage. We should not think of prokaryotes as just disease-causing organisms anymore, because that's not the only thing that they do. Yes, they do cause a lot of human misery and suffering, because they do cause about 50% of all human diseases. But as I hope I've illustrated here, bacteria also can be great collaborators, I guess, because of their biochemical flexibility, because they can make so many different types of organic materials, because they are adaptable and can utilize such a wide range of different chemicals as energy sources. Even ones that we considered to be really, really toxic as human as products of human industrial activity. So dioxins and things like that are considered to be really, really toxic materials. There are bacterial that will break that down. They're more than happy to utilize that as a source of carbon and break it down and neutralize it. And this is only going to increase our use of bacteria, and our understanding of just how flexible bacteria can be in breaking down our industrial waste is only going to expand, and it has to expand because it may be the only logical way that we can eliminate much of the waste that we've placed in the environment. So bacteria, prokaryotes, are also our friends and our collaborators. So one more concept check and then we're done. Bacteria are fundamentally important to the success of other organisms. Identify at least 2 ways bacteria have affected your life positively today. This is one that I'm not going to provide an answer for. This is one that I want you to stop for a moment and just think about where you have encountered bacteria in your life today where they've been beneficial to you. I'll give you 2 for myself, but you come up with some of your own. So I enjoy yogurt, and I know yogurt contains bacteria that are referred to as probiotics because they are bacteria that are found in my gut. And there is a very strong connection between the health of your gut and your physical and mental health. And so I make sure that I both eat yogurt and also take a capsule of probiotic bacteria every day to make sure that the bacterial content of my gut, as demonstrated by research, contains bacteria that are beneficial. So I've read material about how bacteria can influence physical and mental health. And I've looked at the research papers, and I've looked at what products have been tested and have introduced those into what I consume on a regular daily basis. And then if you take vitamins, I don't. I used to once, but I don't take vitamins anymore because I've tried to make my diet as balanced as possible. But if you take a vitamin pill, the chances are that a large number of those vitamins were produced by bacteria. And in fact, some of the bacteria that live in your lower gut, in your large intestine and colon, are producing vitamins for you. Vitamin K particularly is the product of bacteria in your lower gut, and you absorb it through your large intestine. So that would be another way. One other way that I know bacteria have had a positive effect in my life today. I'm human, I produce waste, and bacteria are going to break that down. That's one of their jobs as a decomposer and recycler. But think about how bacteria have affected you today and come up with your own answers. So that's bacteria. And we are done for this one. I believe I will be able to post it in sections so that you don't have to watch it all the way through. You can watch it a section at a time. So until next time, which will be next Tuesday, when we will be looking at the almost as interesting and diverse world of protists. Be safe, and I'll see you then. Bye.